The sensory cells that allow us to hear are called hair cells. Mammals such as ourselves, we cannot regenerate hair cells, whereas in zebrafish and chicken, reptiles and amphibians, these cells regenerate all the time. So they will never lose their hearing. As we age, or for example, as we take chemotherapeutic drugs or noise exposure can kill these hair cells in our inner ears. And as a result, we lose our hearing and also we lose our vestibular system. That's why elderly people have trouble sometimes with their balance. So we're using the zebrafish as a model to study cell proliferation during regeneration of a sensory organ that all aquatic vertebrates have in their skin. The sensory lateral line system. These are sensory organs that allow the fish to detect water motion. They can detect prey, they can detect other fishes. These sensory hair cells in the skin of these fish are very similar to the inner ear hair cells that we possess. There's really an urgent need to understand how these hair cells regenerate in fishes and reptiles and amphibians, because that will help us to hopefully trigger regeneration in mammals in the future. Cells need to proliferate to replace the cells that are dying or being shed. When we kill these hair cells with high doses of antibiotics, for example, or chemotherapeutic drugs that we just add to the water, the hair cells die within 30 minutes, but 5 hours later, already the new hair cells reappear. So we need to understand how proliferation is regulated. How do the stem cells know when they should divide? How do their offspring know whether they should divide? And how do they know at what point they should differentiate? The interesting finding that we made is that there are two different populations of cells that divide. The proliferation of these two different cell populations is independently regulated. There are cells more on the outside of the organ. They divide symmetrically, meaning their two daughter cells are identical. And these two cells actually remain as stem cells. There's a second pool of cells more in the center of the organ. These cells also divide symmetrically, but both of them then form hair cells. And so that allows the organ not to deplete the stem cells, which are on the outside, even though it's continuously making new hair cells. We're collaborating with people working with mice, with chicken, with also zebrafish, and even human tissues. So whatever we learn about the regeneration of hair cells in the lateral line sensory system might also be applicable to other regenerating organs or tissues.